All right, so hello everyone, my name is Johanna M Holman, and I'm a PhD student at the University of Maine, where I work under Dr. Sue Ishak and Dr. Yanyan Lee on a project involving broccoli bioactives, inflammation, and the gut microbiome. And so today I'm here to talk to you about this project and specifically how your gut microbes might think broccoli is delicious. So to start, there's an anti-inflammatory compound that's found in broccoli in a precursor form, and that precursor is converted to the anti-inflammatory by an enzyme when the plant is damaged. But the enzyme is inactivated by high heats, such as in cooking, and since humans don't have the ability to perform this conversion themselves, you would expect not to find that anti-inflammatory in people who have eaten cooked broccoli. But you do. And so the current hypothesis is that gut microbes are performing this conversion from precursor to anti-inflammatory. And there's been a lot of studies looking at this, and this hypothesis is really well supported. So precursors are converted by gut microbiota to anti-inflammatory. Huzzah. And to get a full picture of the gut microbiome, we need to go over the concept of biogeography. And that is this idea that microbes are different across locations, so time and space. So we expect the microbial community in the upper intestines to be very different from the lower intestines, for instance. And that's what you are seeing here in these pie charts. They're differing across the entire gastrointestinal tract of a mouse, so starting with the esophagus through the stomach, and then we have the upper intestine, the mouse cecum, and then the lower intestine. And as you can see, the colors on the pie charts are really changing as you're moving across these locations. And this concept really means that when we're studying gut microbiomes, it's important to try to study specific locations and to not take the entire gastrointestinal tract together. So that's something we really try to do with this study. And so the compound found in broccoli, which is called sulforaphane, is really well studied. And at this point, there's a really large body of work supporting the anti-inflammatory activity of sulforaphane. So that's not a question that we are trying to answer here. What we're interested in is the conversion of the precursor to the anti-inflammatory by the gut microbiome. And we're specifically asking, does a diet of broccoli impact the microbial communities? Who are these gut microbes and how are they performing this conversion? So we took 40 mice, C57BL6 mice, and we performed a pretty well-established mouse model of ulcerative colitis. This is basically the model that everybody uses when they're studying sulforaphane. And we fed the mice either a 10% broccoli sprout diet or a control diet, and then we administered dextran sulfate sodium in the drinking water, and this kind of gives them inflammation. Over the course of 5 days, we gave them this drinking water and they developed inflammation, and then we gave them a break for 5 days, and then we repeated this cycle for a total of 3 times. And after 40 days, we sacrificed the mice and we took tissue and gut content samples, and we also took weights and blood and feces samples, and we assessed them for inflammation and performed 16S rRNA sequencing to take a look at their gut communities. And so this slide has a lot of information on it, but I just wanted to show you that we looked at 4 inflammatory parameters: weight over the course of the entire study. And for this, we expect that mice who have inflammation will gain less weight than mice who don't have inflammation. We looked at disease activity index over the course of the entire study. We looked at lipocalin in the plasma, which is a pretty common inflammatory marker. And we looked at inflammatory cytokines in the plasma as well. And from these, we were able to conclude that, unsurprisingly, mice that were fed broccoli sprout diets were protected from inflammation severity. So they had less severe inflammation. And And so we took a look at 5 anatomical locations for mice. We took gut contents and gut scraping samples, and we did a 16S rRNA analysis on those contents and scrapings. And we found that mice fed a broccoli sprout diet had greater species richness than control mice. And this effect was most strongly seen in the colon. So the colons of these mice had better species richness if they were fed broccoli sprouts. And this was true both of the groups that got DSS and had inflammation and the groups that didn't get DSS and so didn't have inflammation. So a control mouse that didn't have DSS and was fed broccoli sprouts had better or more species richness than a mouse that didn't have inflammation and was fed a control diet. And so we also looked at community clustering according to anatomical location. And what you see in this figure is this control group that didn't have inflammation. What you see in this figure is that they have community clustering by location, as we would expect to see, given that concept of biogeography. So the communities are going to be different in different anatomical locations. And we can also see that this occurred in the control diet group that didn't get DSS and the broccoli diet group that didn't get DSS. And excitingly, the broccoli sprout mice that did get DSS and so therefore had inflammation also had this clustering by anatomical location, while the controlled diet fed mice who had DSS did not have that clustering, and their microbial communities were kind of all over the place. And so this is exciting because this suggests that broccoli is protecting these mice from DSS-induced microbial changes. And so lastly, we looked at putative GLR converters. That's microbes who are predicted to have this ability to convert our precursor to our anti-inflammatory sulforaphane. And we looked for genera that are able to do this conversion. And we also looked for genes that are related to enzymes that have similar activity to the broccoli enzyme. And we did find that genera that are predicted to be able to do this conversion from the precursor to the anti-inflammatory are higher in broccoli-fed mice. So there's more GLR converters in broccoli-fed mice. And that's exciting. However, we actually didn't find that the genes related to this enzymatic activity were uniformly increased in broccoli-fed mice versus control-fed mice. So it's possible that the microbes aren't using this specific enzymatic activity to do this conversion, and we really need to look more at this specific part of the study to resolve this question. And so inflammatory bowel diseases are a rising problem, especially in industrialized nations. We're seeing a lot more diagnoses of inflammatory diseases as time goes on, and diet can be a widely accessible tool for treating and alleviating inflammatory symptoms. So it's important to consider dietary measures, and broccoli is certainly widely available and a well-studied vegetable that contains anti-inflammatories and may protect against inflammation-induced microbial changes and encourage microbial diversity and richness. So in the future, we intend to take isolates that we cultured from gut samples from these mice and test them for precursor converting abilities. We're actually working on that right now. And we're also working on translating this to a human model. And so I'd like to thank our collaborators and our undergraduates and our funding sources for their help on this project.